over, or I skipped over several slides last time, right? And I wanted you to review them on your own. Hopefully you did. Maybe that helped you with today's quiz. Maybe not. I don't know. Right? But this was one of the tables. If you're in my lecture class, we went over this as well. Right? Kind of help you know the different definitions and compare and contrast between these different categories for our organisms. Right? So for each one of them, knowing whether it requires oxygen for its metabolism is important. Knowing if it can grow in the presence of oxygen, and why are we concerned with that? Why are we concerned with oxygen being even present? What can it be for some organisms? It can be toxic. It can be toxic, right? So if they don't have protection, and that's one of the things that you guys are going to read about for homework and test next week, is whether your organism has protection against a toxic form of oxygen, right, that can form, especially when you utilize oxygen, right? So we have this protection because we use oxygen and we live in an oxygen environment. And that's the catalase test that you guys are going to read about for homework and we're going to do next week. So for growth without oxygen, right? So some organisms can grow without utilizing oxygen for their metabolism. So for each one of these, we're going to answer those questions, and we're going to, as you can see, I have it numbered so I remember which way I'm going in my table, right? So we're going to ask the first question of does it require it? Do we have to give it oxygen, or is it going to die if we don't give it to it, right? So for obligate aerobes, which we are, right, do they require oxygen? Absolutely, right? We're this. If we don't have it, we're going to die. What about stricter obligate anaerobes? No, right? Anaerobe means without oxygen, right? Where aerobe means with. There, Stephanie. Are you bailing or are you staying? Okay. We were just starting to go over this table from the last time. Okay. So microaerophiles, do they require oxygen? Yes, but there's a little caveat here, right? How much? Right, less than atmospheric, small amounts of oxygen, right? But they have to have at least a small amount of oxygen. Facultative anaerobes, do they require oxygen? No, right? And this is one where it's tricky, right? Because they don't require it, but they can what? They can use it. They can use it, but they don't require it, right? So saying required knocks them out of this category. Aerotolerant anaerobes. They don't require it. They don't ever use it. Growth in the presence of oxygen. So in other words, do they have protection against oxygen? This isn't whether they use it or not, right? It's whether they can grow in the presence of it, right? So obligate aerobes, can they grow in the presence of oxygen? Yes. Constrict anaerobes. No, oxygen's too toxic, it will kill them. They don't have protection. Microaerophiles. Yes, but again, it has to be small amounts, right? Facultative anaerobes. Can they grow in the presence of oxygen? Yes, they can. And they actually will use it even, right? And grow abundantly. Aerotolerant anaerobes. Can they grow in the presence of oxygen? Yes. Right? Aerotolerant. That part of their name says they can tolerate it, right? They have protection. But do they use it? No. Okay. Can you grow without oxygen? If you're an aerobe, no, we'll die, right? Without oxygen, we will die. We cannot grow without oxygen. Strict or obligate anaerobes, this is the only condition in which they will grow, right? Is without oxygen. Microaerophiles, can they grow without oxygen? No. Remember, they've got to have at least a little bit. If there's none, they're not growing. Facultative anaerobes, can they grow without oxygen? Yes. But slowly, right? Because they can't do aerobic respiration. They got to do fermentation, right? Or anaerobic respiration. Aerotolerant anaerobes, can they grow without oxygen? Yes, they have anaerobe in their name, right? Which means they can grow without oxygen. And in fact, that's always the case, whether oxygen is present or not. The only ones that change between oxygen and non-oxygen are who? The facultative anaerobes. The only ones that can do both, with and without oxygen, and is not affected by oxygen negatively. So we found out with our fluorothioglykylate tubes, our organisms were one type or another as far as their oxygen classification. So what were our two choices for our organisms? 1 and 3. 1 is our obligate aerobes that only grew at the top, right? Or you had a facultative anaerobe, and if you were lucky, you saw more growth at the top, but you definitely saw growth in the bottom of the tube. But why did we do this test? What do we want to know about your organism? Can it grow anaerobically? Can it grow in the bottom of your tube, right? So only you guys that have facultative anaerobes actually had growth in the bottom of the tube. The rest of you were negative for the anaerobic test. They only grew aerobically. So you'll notice that that's how it's worded on your report. And remember, we labeled, right? So no one's going to lose points. So the anaerobic jar, remember we talked about it, but we didn't actually do it. This is the one I wanted to look at. So Pseudomonas, right? If we put that inside the jar under anaerobic conditions, it will not grow. If we leave it outside the jar under aerobic conditions, it will grow. So based on that, what would we classify this one as? This is aerobic, right? A strict or obligate aerobe, just like us. Where Clostridium sporogenes grew inside an anaerobic jar and will not grow outside in aerobic conditions. So this is your anaerobe, your strict anaerobe. Where Staphylococcus aureus will grow inside the jar and outside, but you'll notice significantly more growth outside under aerobic conditions. This is a facultative anaerobe, right? So we could have done this, right? We could have put it in, and again, we can answer the same question. If it grew in that jar, we know it's anaerobic or a facultative anaerobe. But you guys already know you don't have an anaerobe, right? Because most of the time we've got oxygen in this lab and it would kill them. Okay. So that was just a few of them when I wanted to just go over a little bit more. And now I'm going to actually go to 10. And I just cannot. Can't do it. So the first half of the course, we've really been focusing on structure, right? Now we're actually starting to get into what can these organisms do, right? Things like, does it use oxygen for its metabolism? Does it ferment glucose, right? These more functional type questions. So it's kind of like anatomy and physiology, right? And we're finally getting to the physiology. So, you know, we've asked structural questions, right, like capsule, but now we're starting to ask questions like function. Does it ferment glucose, right? And so as you guys saw, all your organisms ferment glucose, right? We all had orange tubes. So today we performed the methyl red and Voges-Proskauer, commonly abbreviated the VP test. And we looked at the results from our Phenol Red Broth test. And as I said, we found out all of ours ferment glucose. Whether they produce gas or not is another thing. So these are what are referred to as differential tests, just like gram stain, right? We could have had one result or another or multiple results. So we've used this term once before when we were talking about how the TTC goes from colorless to red, right? And I hope soon to play around with some new media and actually get us some prettier motility stabs to look at, since mine are like shrunk this small, right? We're like looking at shrunken heads at this point, right? Hoping to prep us some new tubes for next week. So we talk about reduction and oxidation. Commonly, these happen together. They're called redox reactions. And what that is the movement of electrons, right? So a lot of people remember the acronym oil rig. So oxidation is the loss of electrons, where reduction is the gain. Well, if one molecule is losing electrons, another one is gaining it, right? It goes hand in hand, right? So these are redox reactions, reduction and oxidation reactions. And these happen all the time in the cells, right? So the electrons don't disappear, they just go from one place to another. And this is related to what we've been talking about as far as why is oxygen so important, right? Because for aerobic organisms like ourselves, oxygen is the final electron acceptor. It's the last one to accept the electrons that we take away from sugars like glucose, right? So a lot of times, in addition to those electrons, protons will move, or whole hydrogen atoms, right? So a hydrogen atom is a proton and electron, right? And sometimes the whole thing will go together. So we have modified nucleotides like NAD, right? Most of you guys don't even know what NAD stands for, right? But you've heard it, right? You've heard of it as an electron carrier, right? It picks up electrons and carries it from one place to another in a cell. Well, this is actually the chemical formula of it, and it is a modified nucleotide, what makes up DNA and RNA, right? So you have the sugar, the nitrogenous base, and the phosphates. It's actually kind of a dinucleotide. And it'll take electrons from organic molecules like maltase to turn it into oxaloate. When it does that, it picks up electrons and hydrogen atoms. So that's why it goes from a positively charged molecule to a neutral molecule with an additional hydrogen, right? Because that whole hydrogen atom actually moved as well. And this is one of the steps in the Krebs cycle, right? When we break down sugars to get the energy, we're releasing those electrons, and those electrons are what drive the cellular processes. So nicotinamide, right, is what the N stands for, and that's this part of the molecule. And this is an adenine in the other part. So that's why it gets N, A, and then D, because it's a dinucleotide. So it's an acronym. It actually stands for something. So the three main processes that cells will do for respiration is glycolysis, right? So you can take a glucose molecule and you can break it down into two pyruvates. When you do that, and some people will also refer to it as pyruvic acid. You can take that and convert it even further in a gateway step before it enters into the Krebs cycle, right? So we turn those two pyruvates with coenzyme A into two acetyls and two carbon dioxides. The acetyl groups go into the Krebs cycle, right? And they get broken down into 4 molecules of carbon dioxide, right, and regenerates the coenzyme. So we can go back and grab some more pyruvate, right, get them ready to go into the cycle. In that process of breaking down the acetyl group into the carbon dioxide that we exhale, right, a bunch of electrons are released from breaking these bonds between these molecules. So electron carriers like NAD, and you may have heard of FADH, pick up those electrons. That's why they're called electron carriers, and sometimes protons along with them, right, or whole hydrogen atoms. They bring those to the electron transport chain, right, and the electrons, as the chain says, are transported along that chain. And if you're an aerobic organism, what's at the end of that chain? Oxygen. And it accepts the electrons and forms water, what we call metabolic water, because we made it in our bodies. So from glycolysis, the gateway step, and even the Krebs cycle, electrons are stripped away and brought to the electron transport chain. No. Yeah. Okay. So you have glucose, right, which we gave our bacteria glucose, right? And they could have broke that down by glycolysis, right? All carbon all carbohydrates are, or sugars are, carbon and water, right? That's their chemical formula. Here's one of those electron carriers. So as I said, notice it's picking up electrons. You even generate enough energy to add phosphates to ADP, another modified nucleotide that holds energy in those phosphate bonds, right? We've all heard of ATP, right? When you break that bond between the phosphates, you release that energy back. So right now we're storing it, right? We're putting that phosphate on and we're storing it. And we can do that right at the substrate level. Enzymes are doing this, right? We don't need the electron transport chain for that. And then there's the gateway step. So again, as I said, notice electrons, right? And we're even getting rid of some carbon here in the form of carbon dioxide. So now we're going to go into the Krebs cycle, right? So we're going to add it to oxalate, and again, we've got the formation of a molecule, which is why the Krebs cycle has multiple names. This molecule, is, you might hear, is called Tricarboxylic acid. It's because of these three, right, carboxyl groups, and they're acidic in this particular state. And it forms a molecule we know as citrate or citric acid, right? And this is important when you guys are reading for your homework for next week, right? We're going to talk about citrate, right? That's one of the tubes that you guys are a little obsessed with right now, and I don't blame you, right? It's kind of cool. Some of you guys have noticed your green tube, which was our citrate tube, that's the way it started out, has turned what? Blue, right? So as you're reading, right, think about why, right, this is called the citrate test, but what are we talking about as it relates to citrate? What are we doing, right, with the citrate? Okay? So in this case, we form this in the cycle, and then throughout the cycle, we remove electrons. So there again is NAD. And another electron carrier, FAD, right, becomes FADH2. They're grabbing electrons, and then at the end, the carbon is given off as carbon dioxide. And we regenerate the oxalate. That's why it's a cycle, right? It just keeps going, right? We add in the acetyl group, the 2 carbons, right, to the 4 carbon, and it just keeps going around and around. Although things can jump off, right, or come into the cycle. So reduced, right, gained electrons, right? The carbon, on the other hand, has lost electrons. It's been oxidized. So in the citrate test, right, it detects the take-up of citrate from the media, right? So it's not about whether it's synthesizing or making it. It's whether it's taking it in from the media, right? So we put citrate in the media, and we're testing to see if your organism takes it in. Can it take it in and then, process it through the Krebs cycle? So here's the electron transport chain. It are proteins embedded within a membrane. For us, anyone know where that membrane is in our cells? In the mitochondria, right? In the mitochondria is where this membrane is. So these electron carriers carry those electrons to the membrane, and they pass them down along, right, one to the other like a bucket brigade, and at the very end they combine it with hydrogen to form water. In that process, protons are pumped from one side of the membrane to the other. So these are hydrogen ions. They're just the proton part of it. So that's why they're H+, or sometimes just referred to as a proton. That's all they are. So we pump them across. So for NAD, we pump 3 sets. For FAD, oops, I'm going the wrong way. 2 sets, protons across. Why is that important? Because once we pump them across, they want to come back, of course. It's always the way it goes, right? You bail in the boat and the water just keeps coming back in, right? So the protons want to come back, but the only way they can get through the membrane is through this protein port right here. And this protein is a catalytic enzyme called ATP synthase. So it uses the energy, the force of those protons flying back over. So you can almost imagine how water goes over a wheel, right, and can spin that wheel for a mill. The same thing here. This force of those protons going flying through that hole generates enough energy to add phosphates to NAD to form ATP. Right? So this, because it's using electrons, is referred to as, I should have it here, yes, oxidative phosphorylation. All right, so we're adding phosphates because of the power originally of the electrons, right, going down the electron gradient. So, big whip to do, right? You're all like, how much of this do I have to memorize? You don't have to memorize any of it. You have to understand what's going on, right? So, electrons are going down the electron transport. You should already know this from biology class, right? Hopefully some of this is a little bit of a review. Right? Protons go across, they come back, and they're going to form ATP, okay? That's what we do, right? And for us, at the end, is oxygen. And that's why we need it. And if we don't have it, we can't do this, right? So aerobic organisms, you don't give them this system shuts down, right? They can't generate energy because they have no place for the electrons to go, right? This is the problem. The other problem we run into is renegade electrons. Those suckers, man, they just don't want to stay where you want them to go, right? Some of them jump ship, right? So they jump off the electron transport chain, they bind with oxygen, and they form things like superoxide ions. Guess what? Those are bad. They're charged, and they don't know what to do sometimes, and they go interfere with other molecules in chemical reactions, right? This is when I said oxygen can be toxic. Yeah, that's one of the bad guys right there, right? So when we do this, we risk some of them jumping ship and forming these toxic forms. So we've got to have protection against this, right? Because we can't stop it from happening, right? The best we can do is try and protect ourselves, right? So we try to build, higher walls on the causeway because, idiots are going to drive off the side of the bridge anyways, right? So, it's going to happen. So the other one that can form is it can combine with hydrogen and oxygen, and it can form hydrogen peroxide. We've all heard of this, haven't we? You've maybe even used it in your home. Our cells actually make it, right? Sometimes they make it accidentally, right, here where they jump ship from the electron transport chain. Or some of our cells in our body actually purposely make it because it's toxic, right? So when we ingest microbes inside our white blood cells will actually make hydrogen peroxide and pump it into a vesicle where the bacteria are to try and kill them. It's not. Antioxidant is a different type of thing. It's toxic oxygen. Think of it that way, right? This is a toxic form of oxygen. Okay, so guess what? The catalase test. Catalase is an enzyme that breaks hydrogen peroxide into water and safe gaseous oxygen, right? So this is why whenever you pour hydrogen peroxide on a wound, which they tell you not to do now, it should always, always bubble. Because we are aerobic, are we not? Do you think we have catalase? We better. We make hydrogen peroxide all the time. We better be able to deactivate that stuff, right? When we deactivate it with catalase, we produce water and gaseous oxygen. That's why it bubbles. But I'll tell you guys next week why sometimes it doesn't bubble. And it's not because it's not infected. Okay? So this is part of what you will read in your homework. You'll see how all these things are linked together. Okay, so this is the overall formula, right? So we can take carbon like glucose and we can break it down, right? We can remove electrons and we can produce energy right at the substrate level, but most of it is in those electron carriers, right? And those have got to go to the electron transport chain. So remember I said we pump 2 hydrogen ions across for FAD and 3 for NADH. And so because of that, because we pump less hydrogen across and so less come through for FADH, it produces less ATP, right? So FAD produces more. But in general, right, theoretically, you could yield in bacteria, because they don't have to deal with the whole cross the mitochondrial membrane loss thing. They do it right on their cell membrane. They could generate for every 1 molecule of glucose, 38 molecules of ATP. It's pretty good, huh? Right? Most of them come from, and like I said, we got to expend some energy to get across that membrane in the mitochondria. We lose some. Some of them come right at the substrate level, right? And sometimes it's GTP instead of ATP, just a different modified nucleotide. Same process, storing phosphate energy in those phosphate bonds. But most of them come from the electron transport chain. 34 of them, right, come from those electron carriers going to the electron transport chain. And so this would yield quite a bit of energy. So you can even measure it in kilojoules of energy. Actual yield is less than that, right? But quite a bit of energy. So these are some cool YouTube videos that show you like things like the electron transport chain and NADH. So, okay, why did we talk about this? So, what happens if you don't have an electron transport chain? Or in the case of us, which are aerobic, you have no oxygen for those electrons to go. You've got a problem, right? If you're going to break apart these molecules and you're going to release electrons, the electrons have to go somewhere, right? So what do you do if you don't have an electron transport chain? You do anaerobic respiration, right? If you've got something other than oxygen at the end of that electron transport chain to take the electrons, right? But if you don't have that, then the other alternative is fermentation. Yeah, that's where we get the acids, the alcohols, and the gases. Because what we'll actually do is a lot of times somewhere in that path of glycolysis, we're gonna break it down, but we've got to regenerate those electron carriers. So we're actually, what they do is they break down glucose into pyruvate, and then they add the electrons back to it. So they can go and turn around and break down another glucose. They don't use the Krebs cycle, though, because you're going to generate too many electron carriers, right, that have no place to go if you don't have an electron transport chain. So, unfortunately, there's like a gazillion fermentation processes out there. Unfortunate for us, right, because you're like, oh, God, right? But by no means am I going to make you memorize any of them, right? We want to have a general understanding of this. So, in general, because they add the electrons back, we produce things like acids. And sometimes they process it further, they release gases. And they will also sometimes form alcohols. And of course, we love that about fermentation, right? You guys already knew that. You were like, oh yeah, fermentation, alcohol, right? Gotta love it. Right? So in the appendix of your book, there's a whole bunch of different pathways, right? Again, by no means do I want you guys memorizing these. Just to give you an idea of what's happening in these cells at the molecular level, right? So we can take pyruvate, something from glycolysis, right? They can remove carbon dioxide from it. So again, what's carbon dioxide? Gas, right? So that's where some of the gas that we're seeing. They can produce a molecule known as formaldehyde, and then look, they're going to add the electrons back to it and make one of the ones we love, right? An alcohol. Good old ethanol, right? This is the one we drink. This is the one yeast make, like Saccharomyces that we looked at the beginning of the semester, right? That makes bread and beer and wine and all these awesome things. Or we could go this way, right? Again, we're producing some gas. We're producing formaldehyde. We can add a pyruvate to that, and we can form alpha acetolactate. What type of molecule is that? It's an acid. Yep. It's not lactic acid, though. It's a different acid. but similar to what happens in our bodies, and we'll get to that one. Then they can add hydrogen and release some more carbon dioxide here. When they do that, they form a molecule known as acetonin. This one's important. You may have wrote this down in your homework, because we actually tested for the presence of this today. Because what some organisms will do in this fermentation pathway is they will further break this down, right? Or add electrons to it, I should say. This is finally where they're dumping off the electrons. Finally got to that point. They're going to form 2,3-butanol. Which test did we perform today? Were we trying to find out if they produced acetonin? The VP test, right? The Voges-Proskauer test actually tests for acetonin, right? That can be converted, and this is not to be confused, ladies, with acetone, different substance, right? Not the stuff we use to take nail polish off, right? This is different. Right? This is a rate-limiting step, so there usually is enough acetonin in your guys' samples that we can test for the presence of it. Here is lactic acid, right? So this is what we do, right? And you don't want to do this too long because acid is damaging, right? As we'll add the electrons back to the pyruvic acid, right, at the end of glycolysis and make lactic acid, right? But sometimes we want this, right? Lactobacilli make this. This helps make sourdough bread, right? The acids actually are sour to us. Acids are sour. Right. This is also what lowers, there's the same bacteria that lowers the pH in our vagina for women. But this, of course, is not what we want happening in our muscles, right, long term, right, when they're not getting enough oxygenation. We can add the more electron carriers to lactic acid and form propionic acid. So again, notice the theme here, acids, right, lots of acid. Although no organisms actually do it directly like that, they take another path. They form oxalate and then succinate, again more acids, and then finally propionate. All acids, right? So we can also take pyruvic acid, right? Take carbon dioxide from it, form acetyl-CoA. Normally this would enter into the Krebs cycle, right? But if you don't have it, then you've got to do something else, right? Like those pathways we just looked at. Produce some alcohols, produce some gases, right? There's a ton of them, right? Different methods that we can do, but notice the theme. Here's another acid forming. And gases. So the nice thing about our Phenol Red test, right, because Phenol Red is a pH indicator, and it turns yellow in the presence of an acid, what else was in those tubes? Gas. How did we know there was gas in those tubes? Because there was a little tube in there, right? And it trapped the gas, right? The gas displaced the media that was originally in that tube. Now the question is, though, there could be two possible gases. It could be hydrogen gas or carbon dioxide gas. We have no way of knowing, right? It's one or the other, or it could be both that your organism produced. But these are the only two that they produce. Hydrogen gas is the only one that's unique to fermentation, right? Because we expel carbon dioxide when we do aerobic respiration, right? So hydrogen gas is the only one that's unique. Now, some organisms, when they ferment glucose, they're going to produce so much acid that they lower that pH to 4.4 or lower. That's extremely acidic. We refer to that as mixed acid fermentation. So they're not producing just one acid, they're producing multiple acids. And we use the methyl red test to determine that, right? Methyl red, phenol red, too many reds, huh? Drives me crazy. Okay, so as I said, hydrogen gas is unique to fermentation. So this poor dead cow, Clostridium perfringens, which is a spore former that's found in the environment, could get inside this animal, And under those anaerobic conditions, because the animal's dead, it's no longer breathing, it's no longer getting oxygen, it'll grow, and when it grows fermentation-wise, and it produces gas. And that's why the animal, if it doesn't have any large punctures for the gas to release, it will bloat, swell, and then burst, right? So this tissue gas. And some people can get this type of infection as well, right? And the froth will bubble, right, and ooze. It'll literally have bubbles associated with it. And that's because of the gas and the hydrogen gas it produces. The bad thing about hydrogen gas, too, is although it's lighter than air, it's also explosive when ignited, right? And that's what happened with the Hindenburg, right? They had put hydrogen gas in it. Now, what do they put in the blimps or the airships? Helium, yeah, which is non-explosive.
Is it true helium's like becoming rare or something like that, or like?
Yeah, I don't know exactly how helium forms. I don't know if they can make it in the lab or if they get it from the environment. Like, I don't know how it's formed, but yeah, I heard that it was becoming scarce. I definitely did hear that at one time. So this is just a list of several different acids that organisms can produce. And then several different alcohols. And of course, like I said, ethanol is our favorite on this list, but others are also produced that you wouldn't necessarily want to drink, especially methanol. It'll make you blind. And then acetone, a ketone, which can be turned into an acid, can also be produced. And then, as I said, the two gases could be carbon dioxide or hydrogen, but hydrogen is the only one that's unique to fermentation because we produce carbon dioxide. So methanol interferes with the electron transport chain, and so therefore it's would be poisoning. And it affects the eyes first because, believe it or not, it takes a lot of energy to run that whole system of being able to see, to run that optic nerve. So, questions from our homework. Which test for mixed acid fermentation? Methyl red. What reagent did we use? This one's easy. It's the name of the test. Methyl red. And it actually is a pH indicator. That's why we use that reagent. What do the products of this type of fermentation do to the pH? They lower it, right? They lower the pH. And it's really low if it comes out positive for this. So as I said, protons, right, are usually hydrogen ions, hydrogen atoms that's lost their electron. So we measure protons, hydrogen ions, by measuring pH. So again, this is where you just have the proton component. And that's actually what pH is. It's the partial pressure of hydrogen ions within a solution. And this is an inverse relationship. So the more hydrogen ions you have, the lower the pH. Because the measure of it is actually the negative log. So 7 is neutral on the pH scale, right? There's a balance of hydrogen ions and hydronium ions. If it's acidic, so if it's below 7 all the way to even 0, so 4, as we said, 4.4, which is the switch over for methyl red, is definitely acidic. And that means there's a lot of hydrogen ions in that solution. The inverse of that, right, when you go above 7, so a pH of something like 10, is actually less hydrogen ions. So notice, to the minus 10 per mole. So high hydrogen, low pH. Low hydrogen ion concentration, high pH. Also referred to sometimes, basic is also sometimes referred to as alkaline. I don't know why they didn't pick for the coding of our phenol red test, like B for basic. I don't know. So these are a whole bunch of different types of pH indicators. Notice some of the dyes that we use actually will even change in response to pH. So crystal violet, right, will change color in the response of acidic pH. The other ones, of course, that we use are phenol red. And phenol red will turn yellow at not a very low pH. Look at this is 6.8. This is just barely acidic. Right? So think about this. Those of yours that turned yellow in the phenol red test, they might not have turned red on the methyl red test because methyl red is a different range, right? That's very acidic. It has to be down here at 4.4 to be red. Otherwise, it's orange to yellow. So you could be positive for fermentation in the phenol red test because it's way up here on the acid scale and not be positive for methyl red, right? But if yours is methyl red positive, it should also be what? Phenol red positive, right? Because if it's 4.4 here, it should definitely be yellow for the phenol red test. Does that make sense to you guys? So that answers your questions on your report. The other one that we're going to work with next week is bromenthal blue. And this one, we're looking for a change to basic conditions. It's going to actually turn blue. At pH just above neutral pH, 7.6, it's going to turn blue. So some of your tubes have turned blue. But why is that, right? It's one of the questions you'll answer for homework. So the next is our 2,3-butanol fermentation. So which test told us if your organism could do this? This is the Voges-Proskauer or the VP test. We used two different reagents, right? I promise I won't make you spell them on the test, but you do need to know them. We used the first Barrett's reagent A, which is alpha-nethaphenol, and then B, because remember the order matters for these, we used potassium hydroxide. And this is why we needed to wear gloves, because this is a really strong base. You get a chemical burn from it. What did these chemicals interact with? What would be in our media that our organism produced and made via fermentation that we're actually testing for the presence of? Acetonin. You can handle spelling this one, y'all. So what happens is there's glucose in that tube, right? Your organism could ferment it, right, and produce the acetonin that it could later convert it into 2,3-butanol. What we did is we added a reagent, alpha-nethaphenol, which would bind to the acetonin. Then we added the potassium hydroxide, and we had to make sure the second time we added a reagent, right, the potassium hydroxide, we flicked the tube and got bubbles because we needed to add oxygen to this equation. So notice the oxygen right here. Without that, you won't get the production of red color from the diacetyl that forms and the guanine that's in the media that binds and forms a molecule that produces a red color. Right, so positive was red. I don't think anybody got the weird amber color. It's negative. So for both of these tests, a positive result is what? Red, right? This one meaning the pH is 4.4 or lower. This one meaning a chemical reaction happened, right, that produced red color. For some reason, the semester when I took these pictures, it looks more pink. But we've been getting better red. I don't know, I think it was old reagents or something. So, I ran controls, right? Why do we do that? See what it's supposed to look like. What else? What do you think, Rebecca? You're going to say see what it looks like, right? It's good to see what it looks like so you can compare. And also, especially really important next week, to make sure your reagents are correct, right? So we already did this, right? We moved 1 ml into 2 tubes so we could run the 2 different separate tests in 2 little test tubes. Right? And we waited in 10-minute increments, right? I think we might have waited an hour, but I don't know. Kind of cheated a little bit on that one. So one of the questions is, a false negative result for the VP test would more likely be attributed to poor sensitivity or poor specificity of this test. Well, we got to go way back from the beginning of the semester and try and remember these definitions, right? So sensitivity versus specificity, right? So how sensitive a test is versus how specific a test is, right? So is the test we're running specific? Are we specifically just looking for acetonin? And will just acetonin cause a color change? Yes. Yes. So that leaves sensitivity. So what has to happen for us to see the color change? We've got to have acetonin, right? It's going to bind with the reagents, produce a molecule that is red. I don't know about you guys, but my vision is not so great as I get older. What's the likelihood you're going to see a tiny little bit of red? Not likely. If you produce a lot of red, then it'd be really easy to detect. What do you think determines how much red gets produced? How much acetonin is in there? So when it's an amount thing, right, the amount of something that's going to cause this change, it's a sensitivity issue. For this one, you've got to have enough acetonin to get enough of a reaction that you can physically see that red color. If you don't have enough, then you're not going to see the red color, and you may end up with a false negative result. It's not because this test isn't specific. It is specific. Right? So we got to make sure we have enough of this. Typically, you would inoculate these tubes and do this test 2 days later, 48 hours. Ours have been sitting for a couple of weeks, right? 2 weeks. So it should have been a significant amount of time to produce enough acetonin that we got positive results. But again, if we didn't incubate it properly, things like that would affect our results. But I think everybody got positive that was supposed to. So why did we shake the tube after adding the second reagent? We had to actually add oxygen into that solution for that chemical reaction to happen. Okay, for the methyl red test, this is just a pH test. So this one we could read immediately, right? Where the other one we had to wait for a chemical reaction to happen. So this one we added 2 drops of the methyl red. Red is positive, right? And if the red disappeared, right, if it became yellow or orange, it was negative. So organisms like E. coli, when you give them things like glucose, they can produce numerous acids. They can produce lactate, they can produce acetate, formate, they'll even produce ethanol and alcohol, they'll produce carbon dioxide and hydrogen gas, right? And succinate, another acid. So that's a lot of acid, right? And it's enough to significantly drop that pH to 4.4 or lower. So why did we do the methyl red test immediately? Like, you put it in, I was like, is it red? Is it not? Just a pH test, right? It's just pH. pH test happens within seconds. Why did we have to wait for the Voges-Proskauer? What are we waiting for? Well, the oxygen was added into a chemical reaction, right? Do chemical reactions happen like that? No, some of them take time to develop, right? For these molecules to find each other and bond to each other, and for this color to develop as more and more of these red compounds form. So the problem with that is time for a chemical reaction to happen. pH is not really a chemical reaction, it's a measurement. So some microbiologists recommend re-incubating organisms producing methyl red negative results, which actually is what we're doing with Rachel's. We're redoing the test because her tube wasn't cloudy enough. She didn't get enough growth of the bacteria. So she may have not inoculated enough or may not have had enough live organisms, right? So if yours came out negative, right, in this case I was like, I know the result she's supposed to get, right? That's questionable. But even if you didn't know, you can incubate it a little longer, because what's going to happen the longer you incubate it, if you actually have live growing organisms? The more what's going to be produced? The more acid, right? So the likelihood of there being enough acid that you drop that pH real low to 4.4 is greater. Right? Give the organism a little bit more time to produce some more acid. So, right, remember we can apply the scientific method to all the tests we run. It's a good review, right, of the whole test. The last is our phenol red tubes. So these are the pretty ones, right, that we started out with. So we all know the code, right? You guys are masters of the code that we use for these tests now. I think that activity we did probably, really was good and worth it. So we tested, you can buy this base broth, the phenol red with the pH indicator and some proteins in it. With several different sugars, right? So you can test for all kinds of different sugars and sugar alcohols, right? The ones that add an OL are alcohol derivatives of sugars. We tested for glucose, right? Which actually isn't on this list. So in it contains pancreatic digestive casein. This is the white protein in milk. And so that's the protein source. Our formula contains salt, right? So it's isotonically balanced. It contains glucose, which is what they could ferment if they choose to. And phenol red as our pH indicator. And then just water, and the pH is adjusted to around neutral, which is why the tubes start out kind of an orangey red color. So our organisms have two choices: protein or sugar in these tubes. And all of ours chose to eat what? Or ferment what? The sugar. All of ours fermented the sugar. So in what general terms, though, could they do with a sugar, right, when they make it into pyruvate, when they break it down by glycolysis? They could produce what? Acids, alcohols, and gases. Which two can we detect in the phenol red test? Acids and gas. Which one can we not? Alcohols. And in fact, we only run one test that lets us know if an alcohol is produced. What test is that? The VP, right? I know some of you guys got to go. So what's the pH indicator in the media? What is it? It's the name of the test. Phenol red, right? For this one, right, 6.8 will turn it yellow. So just a little acidic, right, will turn it yellow. It will, at 8.2, it'll turn that really bright magenta pink color. Change up my pictures. What color if it's acidic? Did we just say? Acidic. Yellow. Right? Yellow. What color does the media turn if alkaline products are generated? Pink or magenta. Right? It's this real vibrant pink. What is the Durham tube? That's the name of this little tube inside. What does it help us detect? Gas production. We don't know whether it's carbon dioxide or hydrogen, right? But we know it's gas. We know it's got gas, right? That's where your gas comes from too, by the way, y'all. When your microbes in your gut ferment, they produce gas. Give you flatulence. So if glucose is fermented, what happens to the pH? So if we ferment, we're going to produce what? Acid or base? If we ferment a sugar. Acids, right? So what happens to the pH when an acid is produced? It goes down, right? pH goes down. Acids, low pH. Peptone, a mixture of proteins and polypeptides, right, is a component in this media. When they deaminate, when they use the protein as a source, they produce ammonia. Ammonia is a base. And so the pH of this media goes up. Right. So one of their choices is they can remove stuff from amino acids. They can decarboxylate them, or they can deaminate them. They remove the amino group. So some of them will choose to do that instead. You guys don't need to know that. We are not going to go over this, and that's that big table that some of you guys had trouble with, right? And I'm not going to hold it to you guys for quizzing or anything like that. There is one question that I do want to ask you guys, is that when you were thinking about this table, and we didn't do this, we don't do this, our broth comes with the glucose already in it. Some scientists will do a set of control tubes that just have a protein in it. They don't have the sugar. So in that case, right, the only thing they could do if they choose to is eat the protein. Right? So the only result that could be accurate for those tubes is a pink result, if it actually eats the protein. For our tubes, our organism have choices, right? So for most of you guys, and we're waiting to find out on a couple of you guys if it does actually eat the protein after it gets done with the sugar. We know if it ate the sugar, right? What happens if it eats the glucose? It actually ferments it, right? And what color does that tube turn, and why? If it eats the glucose, if it ferments the glucose, it turns yellow, right? The pH goes down because of acidic products, right? So acids are produced from the fermentation of glucose. The pH goes down, it turns yellow. What happens if they ate the protein first instead? Right? It turns pink because the pH goes up. Ammonia is produced, right? High pH, it turns pink. What happens if the bacteria eat all the sugar and then move on to the protein? Right? Orange, or if you let it go long enough, it'll turn pink. Right? But as you guys will notice, it'll be kind of a lighter pink. How are you going to know that? Right? That's the trick. Most of the time we want to know if they produce acid. Right? But some of them, if they run out of sugar, and a couple of these organisms are really quick, within 48 hours, right? If I don't take it out of the fridge, don't take it out of the incubator and put it in the fridge, forget about it, you end up with a pink tube. Right? And if it didn't produce gas, you would have no clue that it ate the sugar first, right? But this is important because you want to know that it chose the sugar first, then moved on to the protein. Does that make sense to you guys? So one of your questions is, what did it eat first? For all of you guys, it's sugar, right? All of us had yellow tubes. Nobody had a protein tube. But for two of you guys, we're going to see, if you've got really hungry bugs, and if they go on to eating the protein, right? And we'll see that color change, and depending on where you catch it, right? If you catch it, you might catch it when the pH has come up to about neutral, so it'll be orange, right? And then if you let it completely go and eat all the protein, it's going to turn pink, right? But it's not going to be the same pink also that you saw for the protein only guys. And remember, for them, right, that bright magenta pink lets you know that they just ate the protein. They didn't do any sugar fermentation. Okay? All right. So we're not worrying about this table, right? I'm not going to test you guys on it or anything like that. The one last question, there's two last questions. I do want to go over these last two. So what if you inoculate a slow-growing fermenter? So I'm telling you he ferments, right? But you come back and the tube is still orange and there's no gas. So you code it minus minus. That's a false negative, right? It's a sensitivity issue too, right? Because if he's growing slow, what is he also producing slow? Acid, right? And you've got to have enough acid to get that color change. So earlier formulations of this didn't have as much glucose. So when you came back 2 days later, it had ate all the sugar, right? And it moved on to the protein. So that you recorded an alkaline result. So what's a way we fix this? Well, obviously we said it had a smaller amount of glucose, right? So we added more sugar, right? What's another thing we could have done? So, right, they're eating the sugar, right? But we come back 2 days later and we don't notice they ate up all the sugar and it went on to the protein. How do we catch them? Well, that's what we've done, is add more sugar. What's another thing we could do instead of adding more sugar? So we incubate for 48 hours, right? Just come back sooner, right? Look at it maybe 24 hours, right? These are the two variables here. The time, right, and how much sugar you're dealing with. That's it. So again, what I did for some of your guys' cultures so we could catch them in the act before they moved on to the protein, after they eliminated all the sugars, I took them out of the incubator and put them in the refrigerator to slow them down. But now we're putting them back in the incubator and seeing what happens.